Hello students, the next point in your syllabus, that is the anatomy of sieve tubes. So there is the in previous video we talk about the translocation of solutes. So this translocation of solutes are mostly takes place through the phloem. So the movement of organic and inorganic solutes from one part of plant to the other plant is called as the translocation. So these translocations are mainly takes place, or translocation of solutes are mainly takes place through this phloem. So we studied the phloem structure. So these phloems is made up of the sieve tubes, sieve plates, phloem parenchyma and companion cells. So you have to see in the figures. In that there is the sieve plates are present, and in between the sieve plates there is the cytoplasmic strands are present. So in the phloem tissues of the stem shows that the outer layer is the cell wall, then inner to the cell wall there is the cytoplasmic strands, and on that cytoplasmic strands there are different mitochondria are present. If we see the single sieve plates, in that sieve plates there is the sieve tubes are present, and surrounding the sieve tubes there is the companion cells are present, and in between there is the at the center there is the vacuole are present. In the vacuoles there is the cytoplasm and solutes are present, and surrounding this vacuole there is the membrane which is called as the tonoplast, and also in that cells there is the nucleus are also present. So you have to see in the single cell sieve plates. In that these sieve plates there are interrupted by pores, and at one side there is the attachment of the companion cells. So we have to see the one by one structure of this phloem. So first in the phloem there is the sieve tubes are present. These tubes are elongated cells, and these are arranged longitudinally end to the end. These sieve tubes have plates at cross walls. जो प्लेट्स को क्रॉसली अटैच रहती है, और these sieve tubes are elongated and arranged longitudinally in the phloems. Then second part is the sieve plates. Those sieve plates bears the pores in it. Then each pore has single strands of cytoplasm extending through it and connecting protoplast of adjoining sieve tubes. This helps the movement of solutes. आप sieve plates का स्ट्रक्चर देख रहे हो, उसमें बहुत सारे पोर्स प्रेजेंट है. सो दिस पोर्स in that there is the single strand of the cytoplasm is present, and to this cytoplasm these pores are connected with this protoplast, or these are connected through the sieve tubes. जो पोर्स है वो पोर्स sieve tubes को कनेक्ट होते हैं, और उसमें जो भी सोल्यूट्स है वो सोल्यूट्स उसमें सोल्यूट्स का ट्रांसलोकेशन हो जाता है. सो ईच sieve tubes has a large central vacuole, and whole sap is rich in protein. A vacuole is surrounded by the thin striated membrane called the tonoplast. sieve tubes हमने देखा है longitudinally उसका elongated cells है. तो these sieve tubes contains the large vacuoles है उसके अंदर, and they are rich in proteins or saps, that is the solutes. then these vacuoles are surrounded by the single or thin membrane, that is the tonoplast. This vacuole is surrounded by the protoplasm and it contains the at least one nucleus, mitochondria, राइबोसोम एक्सेट्रा सेल ऑर्गेनेल्स. फिगर में आप देख सकते हो जो वैक्यूल के जो साइड में जो भी पार्ट है उसमें प्रोटोप्लाज्म है. प्रोटोप्लाज्म में सेल ऑर्गेनेलिस प्रेजेंट है. उसमें न्यूक्लियस रहता है, माइटोकॉन्ड्रियाज है, राइबोसोम्स है, एक्सेट्रा सेल ऑर्गेनेलिस वहाँ प्रोटोप्लाज्म में प्रेजेंट रहती है. देन during the maturations, these nucleus degenerates. Some worker consider that this brings about cessation of functional activity of sieve tubes, while other are of the opinion that activity continues so long as cytoplasm is present. So जो स्ट्रक्चर है, स्ट्रक्चर के बारे में जो अभी साइंटिस्ट है, साइंटिस्ट के दो व्यूज़ यहाँ पे है. कुछ साइंटिस्ट बोलते हैं कि जो न्यूक्लियस है वो न्यूक्लियस कुछ समय बाद वो डिसअपियर हो जाता है और डिसफंक्शनल हो जाता है. दैट इज़ द सीजेशन एक्टिविटी उसे बोलते हैं. सीजेशन एक्टिविटी ऑफ सिव ट्यूब्स. कुछ साइंटिस्ट लोगों का कहना है कि सिव ट्यूब्स की जो एक्टिविटी की वजह से जो न्यूक्लियस है डिसअपियर हो जाता है. तो कुछ साइंटिस्ट बोलते हैं कि जो न्यूक्लियस है वो न्यूक्लियस कंटिन्यूस रहता है. जब तक वह साइटोप्लाज्म प्रेजेंट है तब तक उसमें न्यूक्लियस प्रेजेंट रहता है. सो इन ओल्डर सिव ट्यूब्स टेम्पररी और परमानेंट प्लग्स ऑफ कैलोज डिपॉजिटेड ऑन सिव प्लेट्स. तो जो ओल्ड सिव ट्यूब्स है, दैट सिव ट्यूब्स आर deposited or they can make the plugs of callows. Plugs for the opening or closing of the sieve tubes. There is the plugs of callows are present and they can these callows are placed as a plug for the sieve plates. Then this causes temporary or permanent cessation of functional activity of sieve tubes. जो scientist है, that scientist viewed that these callows plugs are the or they can be causes the cessation activity of the sieve tubes. Cessation activity, that means these degenerates the nucleus. Then nucleus, tonoplast, vacuoles undergoes disintegrations and disappears. So जो cell organelles present in these sieve plates, they will be disintegrate or disappears. So Isu believes that in case of mature sieve tubes, cytoplasm and vacuole becomes one system called mycotoplasm. So the another scientist name, that is Isu, they believes that उनका कहना है कि जो सिव प्लेट्स जब और सिव ट्यूब जब मैच्योर हो जाते हैं तब वहाँ पे जो साइटोप्लाज्म और जो वैक्यूल्स है, these systems they gave the name as a mycotoplasm. So this is regarding the sieve plates and sieve tubes which are play the role in the transportation of the solutes. And last 2-3 points, that is regarding the scientist views for the activity of cessation which is takes place in the sieve plates. Then mechanism of the sieve tubes translocation or mechanism of solute translocation और the mechanism of phloem conduction भी हम उसे बोल सकते हैं. To these explaining the mechanism of solute translocations or sieve tubes or phloem translocations. Various theories are proposed. Some of the theories proposed to explain the short distance translocation, and other theories are explaining the long distance translocation. So this is the two types of translocation according to the location of the translate or transfer. In that there is one is the short distance translocation and another one is the long distance translocation. So for explaining the different short distance translocation, different theories are proposed. In that one is the protoplasmic streaming theory, then transcellular streaming theory, contractile protein theory, then electroosmosis. These are different theories for explaining the short distance translocation. So for today's lecture you have to remind that the translocation of solutes takes place through the phloem, and this phloem there are different parts, that is sieve tubes, sieve plates, then phloem parenchyma and companion cells. In that you have to I have to explain about the sieve tubes and sieve plates. In that sieve plates are the or sieve tubes are the elongated cells, and they are arranged longitudinally and they have crossed with these sieve plates. Then I am explaining about the sieve plates. The sieve plates having the many number of pores, and these pores are has a single strands of cytoplasm extending to these connecting protoplast or adjoining the sieve tubes, and they can takes the movement of the solutes. Also in the sieve tubes there is the large central vacuoles, and these will be rich in the proteins. Then vacuoles are surrounded by the single membrane, that is the tonoplast. Then these vacuoles surrounded by some protoplasm, and they can contain the nucleus, mitochondria, ribosome, etc. cell organelles. Then regarding the nucleus structure, some two groups of scientists are divided, and they are mention their opinions. Some workers says the secession activity of the sieve tubes, in that the nucleus will be disappear, and some workers are says about the if these cytoplasm, or so long cytoplasm is present, their cell organelles are also present in that. And then these sieve tubes are some temporary or permanent plugs, that is the callose plugs, and that will deposited on the sieve plates. These callose plugs are for the functioning, or they can be helps in the movement of solutes. Then these at the time of activities, some cell organelles undergoes disintegrations, and they will be disappeared. And these scientist Isau that believes that these cell organelles like cytoplasm, sieve tubes and vacuoles, they becomes the one system, that is the mycotoplasm. So regarding this is the regarding the structure of phloem. Then the mechanism of sieve tubes translocation, there are two different mechanism. One is the short distance translocation, and second one is the long distance translocation. So for explaining the short distance translocation, different theories are proposed, that is protoplasmic, transcellular, contractile protein streaming theory, and also the electroosmosis. So in next video we talk on these theories regarding the translocation. So now I stop here.